Welcome to my 21st video from my 30-day challenge. In this video, we're going to be speaking about the Monera Kingdom, the unicellular, non-nucleus containing kingdom of life. Hi, my name is Eduardo Hernandez. I am the creator of Emergent Chemical Evolution, the latest theory in the origin of life. I'm also the author of the book by the same title, available in all major online retailers. The key aspect of Monera is that it has such a diversity of bacteria. Well, it has two groups. It has a bacterial group and the archaea group. Now, the difference between these two groups is that archaea are composed of such different types of unicellular organisms that live in such a diversity of environments that are detrimental to most other life forms. They're called extremophiles. And they have membrane structures and different types of means of metabolism where they utilize chemicals that would be poisonous to other life forms, particularly to their integrity and structure. Whereas these archaea type of bacteria thrive there and utilize these chemicals for their metabolic processes. Again, these things would have would have been easier to evolve if these things occurred prior to these processes being completely encapsulated in a specific type of membrane. Now hear me out. Ecosystems would have developed during the viral stage of life where the protein coats of certain viruses would have been more specific to certain environments to protect the contents of the virus, meaning that specific proteins would have evolved to protect these viruses at certain and allow them to become active and replicate at certain environmental conditions, as viruses are prone to do today. They don't start to replicate or release their DNA until certain conditions are met that make these proteins release the contents of said virus. Now, genes only code for proteins. Proteins are the main function and the main cause of function in the different types of membranes, be it organelle, be it the membrane that encases the entire unicellular organisms. Excuse me. These proteins are the basis for all functions, transporting the contents of the environment into the cell and from the cell into the environment, thus giving the function to the membrane. This would have been more easily evolved and differentiated, and the diversity of these would have been much much more, oh, it would have been much more easier to evolve without detrimental consequences during the viral stage, because if the integrity of the membrane would have been detrimentally affected by the introduction of a new type of protein, that would have meant the end for that bacteria. Whereas viruses, their encapsulation would have been affected, but not necessarily detrimentally, and would allow the virus to just maybe wait a little longer, or maybe shorten its period of waiting to get into conditions that would have allowed it to disassemble and reproduce. The exponential replication achieved by viruses would have aided life in its ability to come up with novel and useful proteins. Whereas once these viruses became complex enough to become cellular, that exponential growth as cells became more complex slowed down, and thus became more detrimental to the survival of the cell if changes would occur that were detrimental to its survival by genetic mutation. And again, what's key to realize is that once a specific type of chromosomal conformation is achieved, it would be nearly impossible for organisms to change that system to evolve into another type of chromosomal conformation or arrangement, and therefore becoming a different type of cellular organism. A key thing with Monera is the different types of metabolism that exist in this kingdom. They're autotroph, photoautotrophs that rely on light energy to convert carbon dioxide into useful molecules. They're chemoautotrophs that use chemicals available in the environment to power its metabolism and can garner its carbon from carbon dioxide. There's chemoautotrophs, I believe, that take the energy from light but require to consume other organisms in order to get the carbon it needs. And then there's the mixed autotrophs. Chemotrophs, which are like animals that require complex molecules to power its metabolism and complex carbon sources to eat other animals in order to have carbon to create its molecules. All these 4 different types exist in Monera. They're so genetically different, and their proteins are so different in the jobs that they do, the way they are grouped. And this is key because it points to the core principle of my theory that viroids were the initial common ancestor. Because in my theory, I claim that viroids were the source of both transfer RNA by being autocleaved, and when it was multiplied into ribosomal units. And the way Monera are grouped by scientists is that they're grouped by their ribosomal RNA, specific sequences that match the ribosomal RNA are what they use to group these Monera. And that clearly points to, in the past, there had to be a divergence at this point where the ribosomal, the viroid lineages, where there would be errors would be introduced into the viroids once they were replicated, and then those distinct viroids were replicated to produce ribosomes, and therefore different lineages of ribosomal RNA that would lead to the different species available in Monera. And again, at the viral stage, there will be no Monera, there will be no Protista, there will be no prokaryotes or eukaryotes. It'll all be viral, all viroids, which would eventually lead to the divergent sequences of viroids, which would lead to the divergent sequences of ribosomes, which would lead to divergent types of protein and RNA complexes, which then would produce divergent types of viruses, because then you'll have different types of encapsulation proteins associated with certain ribosomal sequences. And therefore, you would have divergent different types of protein coats associated with specific types of ribosomal sequences. Those ribosomal sequences would then be associated with specific types of membrane types once the viruses became more complex and cellular. Different types of viruses would therefore evolve into different types of cellular organisms, and thereby creating the different types of kingdoms we have now. So anyway, the grouping of Monera by their ribosomal sequences is, to me, another evidence of the validity of my theory. So not only is that a way of unifying all the different types of organisms available in Monera, but that also the existence of Monera and its diversity and the way scientists group them today, which is that the DNA sequences and types of translation and grouping of chromosomal grouping are so divergent in Monera that the only way they can systematize it and organize it was by ribosomal sequences. That, to me, is very telling that my theory is correct. I hope you found this video enjoyable, educational, inspiring. Please like and subscribe. share my videos, and if you care to, purchase my book and read what the fuss is all about. Thank you.